Okay, so let's look at the functions of the DNA. It reproduces itself when a cell divides. So if you look at the process in which the cell divides, makita nyo yung chromosomes natin. No? Meron kang, it becomes visible when a cell divides. Okay. And number two, it supplies the information to make up the ribonucleic acid, okay, RNA. So RNA can make proteins and enzymes as well. So let's look at replication. So I'll do the slideshow here. So in replication, your DNA, if you look at this, okay, so pakita ko lang yung. So this is your original DNA, okay, the green one. So you have two strands here, right? So this opens up, and then you have what you call this first strand is called the lead strand, and then this other one is the lagging strand. Now for the lead strand, the process of making the template. So if you remember, di ba yung mga base pairs niya, G always pairs with C, A is paired with T. So yun, yung kung anong nitrogen bases niya, dun dudugtong sa kabila. Okay? So it will form this duly synthesized leading strand. And then you have this lagging strand. And in the lagging strand, each segment is made separately. So these are called the Okazaki fragments. So unlike the leading strand, in the lagging strand, each part is made up by piece. And then over time, it's stitched together by the enzyme DNA ligase. So in both cases, you have DNA polymerase. But in order for DNA polymerase to work, you need a primer so that it can start its production in making the DNA. Okay, so other important enzymes, you have the helicase, then you have the DNA gyrase. So these are important in replication. Let's go to the next. So replication is bidirectional. It takes place at the same speed in both directions. It is also semi-conservative. Each daughter molecule has one parent strand and one newly synthesized one. So the origin of replication, it's the specific point of DNA where replication begins. And then if you saw a while ago where the two DNA strands separate, this is the specific point of DNA where replication is proceeding. So replication occurs at many places simultaneously along the helix. Okay, so again, the leading strand is synthesized continuously in the 5' to the 3' direction, while in the lagging strand, it is still synthesized from the 5' to the 3' direction, but discontinuously. Okay, so look at the DNA here. This strand is from the 5' to the 3' end. So the other strand is in the opposite direction. It'll be 5' to the 3' end. So your synthesis is both are still 5 prime to 3 prime, but in this case, in the lagging strand case, they are the each segment is produced separately. So you have each fragment of the DNA formed by the complementary bases. Okay, so these each segment that the lagging strand, uh, each segment made on the lagging strand is called the old Okazaki fragment. Uh, I'm still doing a lecture and I am still doing the lecture. I'm not finished. Okay, so you have these assemblies of enzymes that help in replication. We call them replisomes. So you have helicase, which unwinds the double helix, primase, which synthesizes the primers. You have you have the clamp protein, which threads the leading strand, and then you have the DNA polymerase. So it joins the assembled nucleotides, and then you have the ligase, which joins the Okazaki fragments in the lagging strand. Any questions so far? So the helicase is the one responsible for unwinding the DNA double helix. So replication of the DNA starts with the unwinding of the double helix. Unwinding occurs at either end or in the middle. So they attach themselves to one DNA strand and causes the separation of the double helix. So primases are needed to catalyze the primers. So they are short nucleotides. They are required to start the synthesis of both daughter strands. And primases are placed at about every 50 nucleotides in the lagging strand. Another important enzyme is DNA polymerase. It catalyzes the formation of the new strand. So it joins the nucleoside triphosphates found in the nucleus. So you start with the primers that are placed there. So a new phosphodiester bond is formed between the 5' phosphate of the nucleoside triphosphate and the 3' OH group of the DNA strand. So ligase is needed in the formation of the lagging strand. So you have small fragments, which we call Okazaki fragments. They are joined together by the ligase enzyme. Okay, so how do we form the protein? Okay, so let's talk about protein synthesis. So gene expression is the activation of a gene to produce a specific protein. Okay, so yung DNA natin, it's very long, but only a small fraction contains genes. So these are the ones converted into the proteins. Okay. So the base sequence of the gene carries the information to produce one protein molecule. So if you change the sequence, it's an altogether new protein. So this is what you call the central dogma of biochemistry. So DNA replication is when you make copies of new DNA. But when you synthesize the messenger RNA, so from the DNA, you make the messenger RNA, that's called transcription. Okay? Now, from the messenger RNA to the protein, that is called translation. In rare cases, you have viruses that can make DNA DNA fragments from the mRNA. So in the case of HIV, so these are your uh, this can these viruses can do reverse transcription. Okay. So let's go back to when your DNA makes is made into messenger RNA. So genetic information is copied from a gene in the DNA to make a copy to make a messenger RNA. So this begins when the section of a DNA that contains the gene to be copied unwinds. So it unzips inside the nucleus, yung DNA mo, there's an unzipping inside of the DNA. So the RNA polymerase identifies a starting point to begin the messenger RNA synthesis. So here you have the production of your messenger RNA. Okay. So once the DNA splits, you have the template strand, the one here at the bottom, where you make the copies of the messenger RNA, and above it is called the coding or the information strand. So it is not used to synthesize the RNA. So transcription proceeds from the 3' end to the 5' end of the template. So when the messenger RNA is released, the double helix will reform. Okay, so it will seal the DNA again. So in transcription, if this is your base pair sa template strand GAACT, then your complementary base sequence will be CUUGA, diba? So the for G it always partners with C. Pag A, you don't have a thymine in the mRNA, so you have U. Okay, so A always pairs with U in the messenger RNA. So you have C here, it has G. When you have the T in the template, you have A. So this can come up in your exam if you're given the template strand. What are the complementary bases that will form? Okay? So always remember yung mga complementary bases. Lagi mag-partner yung G at saka C, A at saka U sa messenger RNA. Okay? So, thymine, magka-pair naman sa adenine. So, although wala kang thymine sa messenger RNA, yung base pair pa rin niya, it still stands. Kasi adenine naman yung dinadagdag mo sa messenger RNA. Okay, let's try if you get this, no? From the template strand of the DNA below, write out the mRNA and the informational strand of the DNA sequence. So, can you do this series? Yung sa template strand, you have CTA, GATAC. Ano ba itong Okay, so GAUCC, tama ata yan. Okay, so ang mRNA mo is GAUCC-UAUG. Okay? Hindi ko nasusulat yung ano sa exam, no? Basta kabaliktaran pag 3' to 5', sa baba 5' to 3', no? So understood na yun yung direction. Sa informational strand, yung template strand, yung complement niya, di ba? Ganun pa rin. G, A, T, C, C, T, A, T, G. Okay? Kung anong complement dun sa template strand. Another way to do this is just look at the mRNA. Kung saan man may U, palitan mo lang ng T. So that's another way to solve it. Kung kailangan mo ng informational or coding strand. So in translation, the messenger RNA moves out of the nucleus. Meron ka ng messenger RNA and goes to the ribosomes. Now the tRNA converts the information into amino acids. And the amino acids are placed in the proper sequence. And the proteins are synthesized. Okay? So yun yung overall process. Your DNA is you make the RNA. So yun yung transcription. And then yung messenger RNA mo magiging amino acids in translation. So this is the genetic code. So this is the language that relates the series of nucleotides in the messenger RNA to the amino acids specified. So the sequence of the nucleotides in the messenger RNA determines the amino acid sequence for the proteins. Okay? So every 3 nitrogen bases, we call that triplet, this makes up a codon. Okay? So each codon specifies a particular amino acids, and codons are present for all 20 amino acids. So, kumbaga, kung dun sa base pairs natin ay UUUU, that codes for phenylalanine. Okay, so halimbawa, punta tayo sa dati. Yun. So halimbawa, nakita natin dito sa messenger RNA, dito, GAU. GA yung tatlo na yan. So hanapin natin, we use this table. So I want you to print this for the quiz tomorrow or have a copy of this. Okay? So hanapin natin ang GAU, no? So yung titingnan natin dito sa rows muna. So ito yung G. Okay? Tapos yung second letter titingnan natin sa taas. Okay, so dito yung G. Tapos A yung second letter. GAU stands for aspartic acid. Okay? So yun yung amino acid. Okay? So pag UAA, pag UUC, phenylalanine. Okay? So make sure na you understand yung mga, you can use yung ginawa nyo sa amino acids to help to remember ano yung mga amino acids na yan. Okay? So halimbawa, ang code ay GAU, CCU. So yung GAU ay aspartic acid. And then yung CCU, ano ba yung CCU? CCU, titingnan natin. You have CCU. It's proline. Okay? Tapos yung third one. AUG. AUG, ano ba yung AUG? AUG, methionine. So, yun yung magkakabit na amino acids. Gets mo ba, Cerise, how to use the codon table? Para malaman mo kung ano yung nasa mRNA mo, yun yung amino acids na nandun sa protein. Okay? Gets ba? So there are 64 codons that are possible from the triplet combination of A, G, C, and U. And codons are written from the 5' end to the 3' end of the mRNA molecule. So UGA, UAA and UAG are stop signals. Okay. AUG has two roles. It can signal the start of the protein synthesis, and it also specifies the amino acid method. Okay, para matandaan nyo yung stop signals, ito, UAA, UAG, UGA. So imagine a person nasa boat, tapos inuuga yung boat. So tandaan nyo, uga. Okay. So you remember all the stop sequence. So what's the use of the tRNA? So the tRNA translates the codons into the specific amino acids. So it contains around 70 to 90 nucleotides. You have the 3' end, which contains the ACC. So this is the acceptor stem. And this is where the amino acid binds. Okay? And then you have a specific anticodon, which pairs to the codon of the messenger RNA. So if this is UCA, then that means the anticodon will be the complementary base, so AGU. So if you have UCA here, your anticodon should be AGU, so that the carrying molecule, the carrying transfer RNA is specific for serine. Okay, so that's why it's important. So again, the first step, mRNA is made on the DNA, and mRNA leaves the nucleus, attaches to the ribosome, and translation begins. Okay, so eventually you have the different amino acids, no? So halimbawa, to GCC is the codon for alanine. So yung kakabit na tRNA yan will be yung anticodon niya CGG. No? So tuloy-tuloy lang yung production yan. Meron kang ribosomes that are attached to the messenger RNA. So parang factory. Isa-isang transfer RNA. Parang tinataxi nila yung amino acids to the site so that you can produce the mRNA. Okay? So, basahin na ulit natin. So, incoming tRNA, hydrogen bonds to the complementary messenger RNA sequence or the codon. So, as the ribosomes move along the messenger RNA, a new amino acid forms a peptide bond to the growing protein chain. So, the last step is your tRNA re-enters into the cytoplasmic pool of free tRNA ready to be recharged with the new amino acid. So eventually, you form the amino acids you need. Okay, so protein synthesis, the messenger RNA attaches to a smaller subunit of a ribosome, and then the tRNA molecules bring specific amino acids to the mRNA. So peptide bonds form between amino acid and the end of the growing peptide chain. And then the ribosome moves along the mRNA until the end of the codon. So this is translocation. And then the polypeptide chain is released from the ribosome and becomes an active protein. So it will encounter the stop codon, no? UAA, UGA, or UAG. It means it means stop na yung paggawa ng peptide. So there is no tRNA to complement that, so that the enzyme releases the complete polypeptide chain from the ribosomes. And eventually, it will form the 3D structure that forms the protein. Okay? So para repeat lang, no, to re-emphasize. So initiation begins with the messenger RNA binding to the ribosome. So ito yung ribosome mo, yung itong smaller and bigger unit. Tapos, Yung unang ano mo is AUG, which is the start sequence. Also, it signifies it also carries methionine, no? So elongation proceeds as the next tRNA molecule delivers the next amino acid. So yung serine naman, UCA. So kakabit siya sa methionine. So translation continues until stop codon is reached and the completed protein is released. Okay, so eventually you form the completed protein. Sometimes or often the first amino acid is not needed and it is removed after the protein synthesis is completed. So paano nangyari pag nagkaroon mutation sa DNA? So mutation is a heritable change in the DNA. It changes the sequence of amino acids. So what is the consequence? The enzyme cannot catalyze. So what are possible for mutation? This can be X-rays, chemicals like mga toxic chemicals na kinakain, overexposure to sun, viruses. So this can change the DNA. Some mutations are random events, okay? So not all mutation is bad, no? But there are some mutations that can be good for the species, no? So for example, your mutation will result in another color of a flower. So it doesn't mean mag, may mutation automatic disease. Pero usually, mutation can be linked to some diseases. So pag somatic cell yung naapektuhan, non-reproductive cell, so the altered DNA will be limited to the cell and its daughter cells. So this might lead to yung cancerous cells, no? So the cell forgets to stop forgets to die and just keeps on multiplying, dividing itself. Okay, and then you have if the germ cell, like the egg or the sperm, has this new altered DNA, then all new DNA will contain the same default, and it's passed to the next generation. Okay, so let's talk about types of mutation. So meron tayong point mutation. Here you have replacement of one base in the coding strand of the DNA with another. So papalitan mo lang yung isa, halimbawa for G, papalitan niya ng C. So that's an example of point mutation. And then you have the frame shift mutation. One or more bases are added to or deleted from the normal order of bases in the DNA. So all the triplets shift over by one base, and then there's a different sequence of amino acids. Okay? So example of point mutation. What if in your DNA you have CTT, so that codes for GAA. If you look at the codon table, that's glutamic acid. So what if it is changed from T, it's changed to A? So the messenger RNA will be GUA. So instead of GAA, which is glutamic acid, you have GUA, which codes for valine. So iba na yung amino acid dun sa protein mo. So in hemoglobin, the substitution of just one amino acid can result in the fatal disease, sickle cell anemia. So we talked about it. Okay. Another possible situation is instead of CTT, you have ATT. So ATT, if you code that is UAA. So the production of the protein might stop. So instead of glutamic acid, nag-stop yung production. No? So no more amino acids are added. So it may be that the needed protein is not synthesized, and in so doing, the organism may die. So that's how uh, some genetic diseases occur. So we have a certain protein that is not synthesized, that person may die. But sometimes hindi naman ganun ka-fatal. Like for example, yung lactose intolerance, your body cannot produce lactase because of some mutation, or because your genetics doesn't have the gene for the lactase enzyme. So hindi mo madadigest ang lactose. So anong result nun? Sumasakit ang tiyan mo pag uh, uminom ka ng milk or may milk products. So hindi naman siya fatal, but it's very now it will be it will be inconvenient. Siyempre, if you want to drink milk. Another mutation is frame shift mutation. So there are two ways in which you have frame shift mutation. So one is a deletion mutation. So this occurs when one or more nucleotides are lost from a DNA molecule. So instead of yung GAGT-TC, natanggalan ka ng G, so magiging GATTC. Another example is an insertion mutation. So instead of yung magkakaroon ka ng deletion, magkakaroon ka ng insertion dyan. So yung GTTC, magkakaroon ng additional C dun sa in-between. So magka-change yung codons dyan, and it might result in a different protein altogether. Okay? So mas malaki yung effect niyan kasi damay lahat ng mga nitrogen bases. Okay? Another mutation is silent mutation. No? Ito naman, itong silent mutation, nakataon lang na even if you have a point mutation, it will result to the same DNA, same amino acid. So, in effect, hindi siya dangerous, wala siyang outward manifestation. So, the mutation has no effect since even though there's a change um, in the bases, since the codon codes for the same amino acid, then there's no effect. So, yun yung silent mutation. Okay, let's talk about recombinant DNA. So the recombinant DNA is synthetic DNA that contains segments from more than one source. So three key elements are needed to form recombinant DNA. So you have a DNA molecule into which a new DNA segment will be inserted, and you have an enzyme that cleaves DNA at specific locations. So a gene from a second organism will be inserted into the original DNA molecule. So example dyan, in bacteria, you have the plasmid DNA. So what we want to do is we want the bacteria to produce human proteins. For example, insulin, no? So we will trick the bacteria to produce insulin for us. So first, bacterial plasmid DNA is cut by the restriction endonuclease EcoR1. So it's split at a specific site. And then you have the sticky ends where you can insert the gene for a specific protein, for example, for insulin. So this gives a double strand of linear plasmid DNA with two ends ready to bond, called sticky ends. So then a second sample of human DNA is cut with the same echo R1. So yung specific location niya, for example, yun nga yung code for the insulin. So you now have this piece of the genetic code that we can use para insert sa bacterial DNA. So combining the two pieces of DNA with DNA ligase enzyme forms the DNA containing the new segment. So the DNA is slightly larger and the needed protein that we want can be synthesized by the bacteria. So this is how we we produce insulin cheap in a cheap fashion. Kasi dati we had to purify insulin from cows, ganun. So masyadong magastos yung purification and you have to kill a lot of cows to get specific amounts of insulin. So bacteria can be bioengineered to produce the proteins that we need. Okay, another concept here is the polymerase chain reaction or the PCR. So PCR amplifies a specific portion of a DNA molecule, producing millions of exact copies. So first you need the segment of the DNA to be copied, and then you have the two primers. So short polynucleotides that are complementary to the two ends of the segment to be amplified. You also need the DNA polymerase enzyme to catalyze the synthesis of a complementary strand. So nucleoside triphosphates also, these are the source of your adenine, thymine, cytosine, guanine, etc. to make the new DNA. So the first step is you heat the DNA. So that's the first part of the cycle. So in producing heat, you separate the two segments. Step 2, you add primers that are complementary to the DNA sequence at either end of the DNA segment. Okay. And then step 3, you can now use your DNA polymerase to continue the adding of complementary bases. Okay. So after each cycle, the amount of DNA is doubled. No? So you have now two pairs at the first cycle. Tapos you can heat it up again, then mag-split siya, and then the whole cycle repeats itself. So that means after 20 cycles, you can make 1 million copies of DNA. So yan ginagamit sa mga crime scenes. Halimbawa, konti lang yung DNA, but you can amplify the DNA using polymerase chain reaction. So the DNA of each individual person is unique, so DNA can be used as a method of identification. So you can use any type of cell, so kahit yung yung skin, saliva, semen, so this can be used to obtain a DNA fingerprint. So the DNA is first amplified by PCR and then cut into fragments by restriction enzymes. And then you have DNA fragments that are then separated by size by a process called gel electrophoresis. Okay? So, yung DNA mo, pinaparami mo muna, tapos it can be separated into fragments. So, by the molecular weight of each segment, no, magse-separate siya through electrophoresis. And then you have these bands that can be seen. So, ganyan, yung sa DNA fingerprinting, for example, may mga suspect sa crime scene. So, tatry mo ipantay kung sino yung tao na with similar bands. So, that's how they capture the suspect in a crime scene. Okay, we have this last topic. So a virus is a infectious agent. We already discussed this already noon sa micropara. Okay. So, so it's we can move forward with this. So it is incapable of replicating alone. So it invades a host organism and makes the host replicate the virus. So many diseases like cold, influenza are viral in origin. So yan yung yung retrovirus yung pinag-usapan natin. This can do the reverse transcription. From the mRNA, it can make DNA and insert it to the host. Okay? So yun yung reverse transcription. So the viral DNA can then transcribe RNA, which then directs protein synthesis to infect other cells. So yun yung retrovirus. So that's what AIDS, the disease does to our body. So yung HIV will target your T cells, and eventually it will make our body unable to fight diseases that are normally fought off by our immune system. Okay, yung mga types of COVID-19 vaccines, you have the live attenuated. So this is a harmless germ told to make pieces of another germ. So example yung sa AstraZeneca, you have chimpanzee cold virus, tapos it is it is trained to make uh, copies of the protein spikes from COVID-19. So this is live attenuated. So the germ is still alive and can replicate, which will draw attention from immune system and create a response. Pag inactivated, pinatay mo yung virus, for example, that's inactivated. So the germ cannot replicate. So in this case, you need booster shots. So you also have adjuvants. So adjuvants are molecules that are paired with the germ in a vaccine to cause greater immune responses. So they are useful in vaccines to have small concentrations or immunogenicity. You also have the subunit type. So you just have the protein spike. So this is a piece of germ that causes the strongest response. So the piece might be included on a particle and adjuvants may be included. So yung protein spikes na pinag-usapan natin sa virus, that's an example. Lastly, you have the DNA or RNA. You have instructions for the body to make the antigen itself. So adjuvants may be included here. Okay. Lastly, we have the antibody-dependent enhancement. I have to discuss this, no, kasi very we need to this knowledge para pag-aralan yung effects sa vaccine. So we all know this already. So not all antibody responses are created equal. Sometimes antibodies do not prevent cell entry, but they may actually increase the ability of a virus to enter cells. So imbis na nag-help yung antibody, siya yung tumutulong na palakasin yung effect ng disease. So ito yung tinatawag na antibody-dependent enhancement. So this occurs when the antibody generated during an immune response recognize and bind to a pathogen, but they are unable to prevent infection. So para siyang Trojan horse. And this allows the pathogen to get into cells and exacerbate the immune response. So when a vaccinated person gets infected, iba-ibang scenario ang pwede mangyari pag na-vaccinate ang person, tapos nagkasakit. So there are three scenarios. One is, it could be just a mild illness. So the person may experience some symptoms, but are more of an inconvenience and it may last only a few days. No? So common yan sa mga Johnson and Johnson, I think sa Pfizer. So these mild symptoms are evidence that the vaccine has worked. Okay, for some people. And then there is a case na hindi enough yung tulong ng vaccine. So this is called the breakthrough illness. So in this case, the vaccine may not have worked at all, or it did not induce high enough levels of immunity to effectively stop infection. So yung last scenario, yung ADE. So the antibodies that the vaccine generated actually helped the virus infect greater number of cells that it would have on its own. So in this situation, the antibodies bind to the virus and help it more easily to get into cells than it would on its own, no? So imbis na mas na naging severe yung disease. Okay? So this can occur after the disease and has on occasion been identified following vaccination. So any vaccine that has been found to cause ADE has been stopped, or more recently for the dengue virus, recommended only for those not affected by ADE. So yung mga tinatawag natin zero positive, okay lang yung Dengvax siya. But for seronegative, binawal na rin ng mismong company because evidence of ADE has emerged for that type of vaccine. Pero for COVID-19, wala namang evidence ng ADE. So for the most part, the vaccines for COVID-19 is safe. Okay, so that's it. So i-upload ko na lang tong video na to. Okay, so that's all. So thanks for listening. So see you tomorrow. May quiz na tayo for nucleic acid. Okay? Okay, bye. Bye.
Bye po, sir. Thank you po.